In the name of Allah, who is most beneficent and merciful. Assalamu alaikum. My name is Umayya. My ID is 014. I'm from Chemistry Department. And today, topic of my presentation is Laccase, Catalase, Cellulase, and Protease. First of all, you can see contents of my presentation, which include Laccase. In Laccase, I'll introduce Laccase, its synthesis by fermentation, and then I'll discuss measurement of activity of Laccase, then applications in industry and in biology of Laccase. In Catalase, I'll introduce Catalase, then its chemistry, its biological reactions, its measurement of activity, then applications of Catalase. After Catalase, I'll discuss about Cellulase, its introduction, chemistry, types of Cellulase, synthesis by fermentation of Cellulase, and applications of Cellulase. And then at last, I will discuss about Protease, its introduction, chemistry, enzymatic reactions of Protease, and applications of Protease. So let's start with introduction of Laccase. Laccase, which belong to the enzyme family of multi-copper oxidases, are classified as benzene diol oxygen reductases. They are considered versatile enzymes capable of oxidizing a large number of phenolic and non-phenolic molecules due to their low substrate specificity, using oxygen as electron acceptor and generating water as a byproduct. Laccases are widely expressed in nature because they can be obtained from various fungi, plants, bacteria, lichen, and insects. Here you can see basic information about chemistry of Laccase. The structure you can see, the molecular formula, which is C44H69 N11O20. Optimum pH is between 5.5 to 7.5. Now we'll see its synthesis by fermentation. These fermentation techniques are used in the production of Laccase. First is submerged and the second one is solid state mode. But at first I'll discuss about submerged fermentation. Submerged fermentation involves the nurturing of microorganisms in high oxygen concentrated liquid nutrient medium. Viscosity of broth is the major problem associated with the fungal submerged fermentations. When fungal cell grows, mycelium is formed, which hinders impeller action due to this limitation occurring in oxygen and mass transfer. For dealing with this problem, different strategies have been employed. Bioreactor operates in continuous manner for obtaining high efficiency, and two scientists also reported that continuous Laccase production takes place without enzyme deactivation for a period of 4 months due to immobilization of neurospora on membrane. For bioremediation of pentachlorophenol and 2,4-dichlorophenol, nylon mesh is used for comparing the free cell culture of T. versicolor with immobilized cultures. Some scientists also have investigated that in fixed bed bioreactors, stainless steel showed the highest Laccase activity among different synthetic materials which were used as carriers in the fermentation. So the most effective way of producing Laccase is fed batch operating, through which the highest Laccase activity can be obtained. But now I'll discuss solid state fermentation for the production of Laccase. SSF, which stands for solid state fermentation, is suitable for the production of enzymes by using natural substrates such as agricultural residues, because they mimic the conditions under which the fungi grow naturally. The lignin, cellulose, and hemicelluloses are rich in sugar and promote fungal growth in fermenter and make the process more economical. The major drawback is that the bioreactor design in which heat and mass transfer it is limited. Different bioreactor configurations have been studied for Laccase production such as immersion configuration, expanded bed, tray, inert, and non-inert support in which tray configuration gave the best response. A tray and immersion configuration is compared for Laccase production by using grape seeds and orange peel as substrate. Laccase production by both type of fermentations, solid state and submerged, is higher in case of rice bran than other substrates. So the rice bran inductive capability is based on the phenolic compounds such as vanillic acid, which induce the Laccase production. Many agricultural wastes such as grape seeds, grape stalks, barley bran, cotton stalk, molasses wastewater, and wheat bran are also used as substrate for Laccase production. However, Laccase production in both types of fermentation didn't reach up to maximum level. I have told you already that fed batch fermentation is best process for the production of Laccase. Then I'll discuss measurement of activity of Laccase. For determination of Laccase activities, you mix your enzyme solution with an ABTS. An ABTS concentration of the mixture should be 0.2 millimeter. Then you measure absorbance over time at 420 nanometer. Now we'll discuss applications. How it is used in industry. Textile industry utilizes large volume of water and chemicals for wet processing. These chemicals range from inorganic compounds to organic compounds. The chemical structure of dyes provides a resistance to fading when exposed to light, water, and other chemicals. Laccase degrades dye. That's why Laccase-based processes have been developed, which include synthetic dyes and are being used in industries nowadays. Chlorine and oxygen-based chemical oxidants are also used for the separation and degradation of lignin, which is required for the preparation of paper at industrial level. But some problems such as recycling, cost, and toxicity remain unsolved. However, in the existing bleaching processes, LMS could be easily implemented because it leads to a partial replacement of chlorine dioxide in pulp. In food industry, Laccase is used for the elimination of undesirable phenolic compound in baking, juice processing, wine stabilization, and bioremediation of wastewater. Laccase improve not only the functionality but also the sensory properties is. In beer industry, it is also used. Why it is used? Not only provides stability to the beer, but also increases the shelf life of beer. In beer, haze formation takes place, which is stimulated by the naturally present polyphenol and is referred to as chill haze. At room temperature or above, warming of beer can dissolve the complex. So the question is how Laccase increase the shelf life of beer. After a certain period of time, phenolic rings are replaced by the sulfide group and permanent haze is formed which can't be redissolved. For polyphenol oxidation, Laccase has been used, which is capable of removing the excess oxygen and also due to which the shelf life of beer increases with the help of the Laccase. It is also used in bioremediation and biodegradation. Due to rapid industrialization and extensive use of pesticides for better agricultural productivity, contamination of soil, water, and air take place, which is a serious environmental problem of today. polychlorinated biphenyls, benzene, toluene, ethylbenzene, xylene, polycyclic aromatic hydrocarbons, pentachlorophenol, 1,1,1-trichloro-2,2-bis-ethane, and trinitrotoluene. All these are the substances which are known for their carcinogenic as well as mutagenic effect and are persistent in the environment. Fungi innovate a wide variety of hazardous chemicals. That's why the researchers' interest is generated in them. Laccase has the ability to decrease odor arising from the garbage disposal sites, livestock farms, and pulp mills. Since lackases catalyze the electron transfer reactions without additional cofactors, they can also be used as biosensors. Why? Because they can detect various phenolic compounds and oxygen. So these were the applications of laccase. Now moving towards, I'll discuss about catalase. So let's start catalase with its introduction. Catalase is a common enzyme found in nearly all living organisms exposed to oxygen, which catalyzes the decomposition of hydrogen peroxide to water and oxygen. It is a very important enzyme in protecting the cell from oxidative damage by reactive oxygen species. Catalase has one of the highest turnover numbers of all enzymes. One catalase molecule can convert millions of hydrogen peroxide molecules to water and oxygen each second. So now there is basic information of its chemistry structure. You can see molecular formula is 2H2O2. Optimum pH is 6.8 to 7.0. Now we'll discuss about biological reactions. The presence of catalase in a microbial or tissue sample can be demonstrated by adding hydrogen peroxide and observing the reaction. The production of oxygen can be seen by the fermentation of bubbles. This easy test, which can be seen with the naked eye without the help of instrumentation, is possible because catalase has a very high specific activity which produces a detectable response, as well as the fact is that one of the product is a gas. In the reaction, you can see hydrogen peroxide is being converted in molecules of water and in oxygen gas, while complete mechanism of catalase is not currently known. The reaction is believed to occur in given two stages. So now I'll talk about these two stages. Here iron A represents the iron center of the heme group attached to the enzyme. Iron with oxidation state 4 is mesmeric form of iron in oxidation state with 5, meaning the iron is not completely oxidized to +5 oxidation state, so but receives some stabilizing electron density from the heme ligand, which is then shown as a radical cation. As hydrogen peroxide enters the active site, it interacts with the amino acids ASN148 and HIS75, causing a proton to transfer between oxygen atoms. The free oxygen atom coordinates and then iron, which is connected with oxygen with the help of the double bond, reacts with a second hydrogen peroxide molecule to form iron with oxidation state 3 and produce water and oxygen. Catalase can also catalyze the oxidation by hydrogen peroxide of various metabolites and toxins, including formaldehyde, formic acid, phenols, acetaldehyde, and alcohols. It does so according to the following reaction. This reaction you can see there is an acceptor and hydrogen peroxide. The product is molecules of water and R is then separated. The exact mechanism of this reaction is also not known yet. So now we'll talk about measurement of activity of this enzyme. In 1870, scientists discovered a formation of yellow color from the interaction of hydrogen peroxide with molybdenate. Then from the middle of 20th century, This reaction began to be used for calorimetric determination of unreacted hydrogen peroxide in the catalase activity. Now, these are the applications of this enzyme. In bacterial identification, so how it can be used in bacterial identification. The catalase test is one of the three main tests used by microbiologists to identify species of bacteria. If the bacteria possess catalase, when a small amount of bacterial isolate is added to hydrogen peroxide, bubbles of oxygen are observed. The catalase test is done by placing a drop of hydrogen peroxide on microscopic slide. An applicator stick is attached to colony and the tip is then smeared onto the hydrogen peroxide drop. If the mixture produces bubbles or froth, the organism is said to be catalase positive. Staphylococcus and Micrococcus are catalase positive. Other catalase positive organisms also include Listeria, Yersinia, Protease, Salmonella, Pseudomonas, Mycobacterium, tuberculosis, aspergillus, etc. But if not, then organism is said to be catalase negative. And examples are Streptococcus and Enterococcus or Enterococci. Second application is gray hair. Low levels of catalase may play a role in the graying process of a human hair. Hydrogen peroxide is naturally produced by the body and broken down by catalase. If catalase levels decline, hydrogen peroxide cannot be broken down so well. The hydrogen peroxide interferes with the production of melanin, the pigment that gives hair its color. So these were the applications of catalase. Now we'll discuss about cellulase enzyme. Cellulase is any of several enzymes produced chiefly by fungi and bacteria that catalyze cellulolysis, the decomposition of cellulose, and of some related polysaccharides. The name is also used for many naturally occurring mixtures or complexes of various such as enzymes that act serially or synergistically to decompose cellulosic material. Now you can see basic information of its chemistry structure. You can see molecular formula is C18H32O16. Optimum pH is 5.0. And now we'll talk about its types. There are 5 general types of cellulases, and these types are based upon reaction catalyzed. So the first one is endocellulases, which randomly cleave internal bonds at amorphous sites that create new chain ends. The second one is exocellulases, which cleave 2 to 4 units from the ends of the exposed chains produced by endocellulases, resulting in tetrasaccharides or disaccharides such as cellobules. Exocellulases are further classified into 2 types. Type 1 that work processively from the reducing end, and type 2 that work processively from the non-reducing end. So the third one is cellobiases, which the exocellulases product into individual monosaccharides. The fourth one is oxidative cellulases, which depolymerize cellulose by radical reactions. The fifth one is cellulose phosphorylases, which depolymerize cellulose using phosphates instead of water molecules. Now we'll discuss about its synthesis by submerged and solid state fermentation. So first of all, we'll talk about materials and methods. Microorganism. The fungus is isolated from core retting ground area. The isolated fungal colony was subcultured. Pre-treatment of substrates. How pre-treatment is done? Basically, the procured cellulosic substrates such as rice husk, rice bran, coir waste, Wheat bran and sawdust are ground to fine powder, and substrates were added in ratio of 1:10 for 1 hour and was brought to neutral pH by washing thoroughly with distilled water and dried at room temperature. The treated substrates were autoclaved at 121 degrees Celsius temperature for a time of 1 hour. Then preparation of in column. The in column is prepared by growing the organism in 250 milliliter flask with 100 milliliter and incubated at 30 degrees Celsius temperature for a time of 3 days in a shaker before it was used for fermentation process. So then submerged fermentation takes place and it is carried out in 250 milliliter flasks containing 100 milliliter of fermentation medium. The condition for this fermentation is 121 degrees Celsius and 15 minutes. Each flask was inoculated with 1 milliliter of above said in column. The cultures were incubated on a rotatory shaker at 30 degrees Celsius temperature for the time of 72 hours. So what happens in solid state fermentation? It is carried out in 250 milliliter flasks that contain 10 gram of coir waste and 15 milliliter of distilled water. The flasks were sterilized at 121 degrees Celsius for 15 minutes and cooled to room temperature. About 1 milliliter in column was added, mixed well and incubated at 30 degrees Celsius in a humidified incubator for 96 hours. The flasks were periodically mixed by gentle shaking. At the end, enzyme is extracted. At the end of the fermentation, the culture broth from the submerged fermentation was centrifuged at 6000 rpm for 15 minutes and the supernatant was used as a source of extracellular enzyme. In solid state fermentation, the enzyme was extracted from the coir waste by mixing homogeneously the entire waste with distilled water and agitated on a rotatory shaker at 30 degrees Celsius with a contact time of 1 hour. Dampened cheesecloth was used to filter the extract and pooled extracts were centrifuged at 6000 rpm for a time of 15 minutes and the clear supernatant was used as source of extracellular enzyme. Then there are applications of cellulase. It is used for commercial food processing in coffee. It performs hydrolysis of cellulose during drying of beans. Furthermore, cellulases are widely used in textile industry and in laundry detergents. They have also been used in the pulp and paper industry for various purposes. And they are even used for pharmaceutical applications. Cellulase is also used in the fermentation of biomass into biofuels, although this process is relatively experimental at present. Medically, cellulose is also used. How it is used medically? It is used as a treatment of phytobezoars. And the second use in medical field is that it can hydrolyze beta-1,4 glycosidic linkages within the structural matrix exopolysaccharides of the extracellular polymeric substances. So these were the applications of cellulase and fermentation processes by which we can make cellulase I have already discussed. So now at last I'll discuss about protease. A protease is an enzyme that catalyzes proteolysis, breaking down proteins into smaller polypeptides or single amino acids and spurring the formation of new protein products. They do by cleaving the peptide bonds within proteins by hydrolysis, a reaction where water breaks bonds. Proteases are involved in many biological functions, including digestion of ingested proteins, protein catabolism. So basic information is here. This is the structure of the enzyme. Molecular formula is C33, H46, and 807. And optimum pH is 7 to 12 for this enzyme. Now we'll discuss about enzymatic function and mechanism. Proteases are involved in digesting long protein chains into shorter fragments by splitting the peptide bonds that link amino acid residues. Some detach the terminal amino acids from the protein chain. Others attack internal peptide bonds of a protein. Catalysis. Catalysis can be achieved by two mechanisms. Aspartic, glutamic, and proteases activate a water molecule which performs a nucleophilic attack on the peptide bond to hydrolyze it. Serine and cysteine proteases use a nucleophilic residue. That residue performs a nucleophilic attack to covalently link the protease to substrate protein, releasing the first half of the product. This covalent acyl enzyme intermediate is then hydrolyzed by activated water to complete catalysis by releasing the second half of the product and regenerating the free enzyme. And this whole mechanism that I have told you, you can see in these two reactions. When enzyme reacts with substrate in first step of the catalysis, it gives enzyme and products 1 and 2, but then producing intermediate acyl enzyme, it free up the enzyme and produces two products. Now we'll talk about specificity of the enzyme. Proteolysis can be highly promiscuous, such that a wide range of protein substrates are hydrolyzed. This is the case for digestive enzymes such as trypsin, which have to be able to cleave the array of the proteins ingested into smaller peptide fragments. Then degradation and autolysis of proteases. Proteases, being themselves proteins, are cleaved by other protease molecules, sometimes of the same activity. This acts as a method of regulation of protease activity. Some proteases are less active after autolysis, while others are more active. For example, trypsinogen. Now, at last, we'll discuss applications. The field of protease research is enormous. Since 2004, approximately 8,000 papers related to this field were published each year. Proteases are used in industry, medicine, and as a basic biological research tool. Digestive proteases are part of many laundry detergents and are also used extensively in the bread industry, in bread improver. A variety of proteases are used medically, both for their native function and for completely artificial functions. Highly specific proteases such as TEV protease and thrombin are completely used to cleave fusion proteins and affinity tags in a controlled fashion. So these were the applications of proteases, which I have explained briefly. These are the references from which I have got all the material for my presentation. So this was the presentation in which I discussed about different enzymes like protease, cellulase, catalase, laccase, about their activities, their chemistries, and their introductions. That's all from my side. Thank you.